Hi, welcome. My name is Jeri Francoeur. I am on the Alamo Breast Cancer Foundation San Antonio Breast Cancer Symposium Planning Committee, and I want to thank our sponsors now. We have Exact Sciences, Genentech, Sanofi Genzyme, GE, Pfizer, Viatris, Seagen, Daiichi Sankyo, Eisai, Merck, Grail, Sino, Agendian, and Cybermedia Productions. Without them, this could not be possible, so we really appreciate all they do to help us. To get into the introductions of our guests here, we have Dr. Jenny Chang on the far end, Dr. Virginia Kaklamani, and Dr. Help me with your first name.
Olopade, Fumi
Dr. Olopade. And they're going to talk about some of the highlights during the sessions the last few days. So Dr. Chang is going to start off.
Hello, everybody. My name is Jenny Chang. I'm from the Dr. Mary and Ron Neal Cancer Center in Houston Methodist. I'm a breast oncologist and the director of the Cancer Center. Very briefly, first and foremost, I would like to take, this is the highlight of the meeting for me, coming here every year for the past 20 years. Shh. And truthfully, thank you very much for everybody who has actually made it in person, everybody online. It truly is a phenomenal time that we can actually brainstorm, think about what this week has meant to us and what the highlights are, and to answer any questions that you may have, because this is why we wake up every day, is to basically translate what we find. And this year especially, I would actually really like to thank the organizers, Dr. Kaklamani and Dr. Artiaga, for an outstanding piece of work. It has been really, really so difficult. Everybody's been stretched and challenged. And I think it has gone very smoothly, and thank you very much again. I think that we really miss, all of us, we would say unreservedly, we really, really miss San Antonio Breast Conference, and we're glad we're here. So what has this what has this session been? I think it's been a phenomenal how much discovery there is. We kicked off by looking at triple-negative breast cancer, understanding that now we have new therapies for triple-negative breast cancer, approved therapies, especially immunotherapy, but not all triple-negative breast cancers are alike. But when we apply these new technologies that we have by imaging cytoph, by looking at single-cell spatial analysis, we can now understand that the tumor needs an environment. And that environment is maybe what determines whether or not we respond to treatments. And so with that, new treatments that we have with triple-negative breast cancer, knowing that it's not just the tumor, but also the microenvironment, and moving forward with new treatments for that. Quickly forwarding to a session that I moderated, which is basically on triple positive, from triple negative, now triple positive breast cancer. Well, that is a totally different beast, and things that we thought we knew of in the past, it's being turned on the head. It's a unique entity in itself. It has different drivers, and we are now learning new ways. And to be honest, we now have to subset all the different types of tumors because of the heterogeneity that we see within what we deem as HR positive or triple negative or HER2 positive. We now know there's many, many different subsets. And it's really, really fun because we now have phenomenal toys. When Dr. Fumi and I started, I'm sure she remembers that, even Virginia, that we basically had, it took us years just to learn how to measure ER. Now we can do 40, 50 proteins on one slide. It's just a changed world, and we can understand so much more. And with that understanding, it's been translated into, frankly, new discoveries, better outcomes, and changing this dreaded disease into a chronic disease, even if you have stage 4. And I think we're very close, and I think everybody shares the enthusiasm as we move forward. It's no longer 20 years ago, HER2 breast cancer, it was not good. And now we are moving forward to new ways of treating all these by these new tools that we have.
Dr. Kaklamani?
So my name is Virginia Kaklamani. I'm local. I work at UT Health in San Antonio. I've been with ABCF, I think, since 2006 or 7, Sandy. The ladies recruited me when I was coming down visiting from Chicago, and we've kind of worked together since. So thank you. This is my highlight as well, Jenny. I always say that this is the best part of my meeting.
It is.
So what I will focus on today is two of the first presentations that took place during the general session, and both of them had to do with HER2-positive breast cancer. The first one was the Destiny Breast 03 trial, which I think if we look at this year's clinical trials, is probably the one that will be quoted the most because of how impressive the results were. Now, the results were initially presented at a European meeting a couple of months ago, and they showed that this novel antibody drug conjugate did outperform the standard of care treatment, which is called TDM1 or Kadcyla. Now, these antibody drug conjugates are really smart molecules. It's an antibody, and in this case, it's basically Herceptin, and they put a toxin on it. And the way these molecules work is they enter the cell, and then they fool the cell because the cell doesn't realize that they also have a toxin on it. And then that toxin gets released and kills the cell. So we call them the Trojan horse approach because that's how the Athenians beat the Trojans during the Trojan War. So this molecule, this new antibody drug conjugate, which we have approved and we've been using for breast cancer for the past couple of years, now showed that it is extremely active in earlier stages of HER2-positive metastatic disease. It also showed that it's relatively safe. One of the concerns with this molecule is that it causes this what's called interstitial lung disease, basically lung toxicity. And we were all worried about that because obviously we don't want our patients dying from side effects from our medications. But this trial showed that the rates were not as high as we thought they would be, and most people actually did pretty well. And the efficacy of this drug was exceptional. And so the data that was presented today focused on women that had brain metastases. And one of the concerns with these drugs is because they're big molecules, it's very hard for them to go into the brain. And what the trial showed was that they do go into the brain, and they improve outcomes in our patients that have HER2-positive metastatic breast cancer and also brain metastases. So I think this is extremely applicable in clinic. This helps us not be concerned about using this drug in patients with brain metastases, and obviously it's one of our new standard of care approaches for HER2-positive disease. And this drug will move on to the early stage setting in trying to prevent metastatic disease. The second trial was a trial that was done in China with another anti-HER2 drug, which is called Pyrotinib. Now, this drug is not available in the United States. We've seen trials with it, and actually this symposium received many abstracts on Pyrotinib. And the reason we wanted to highlight this one was because it was a study showing that compared to what they use in China as standard of care therapy, Pyrotinib improved overall survival. Again, we don't have this drug in the US, so why do we care? Well, we care because the rest of the world is not just the US. And we are lucky to have many drugs approved for HER2-positive breast cancer, but China doesn't. So in not just China, Japan doesn't have all the drugs that we have. Most of the world doesn't have the luxury to have the drugs that we have. And so Pyrotinib is an option for our Chinese patients, and actually it's used in the second-line setting. And again, it showed activity and improvement in overall survival, and it's actually a pretty easy drug to give. It doesn't have a lot of side effects. And I think it highlights how different countries and different regions treat patients differently, and we have to all take that into account when we talk to our patients. So that's where I'll stop, and we'll take questions at the end.
Dr. Olopade?
Yeah. So again, thank you very much for bringing us to walk and speak to the advocates who are here. I want to acknowledge so many people in the audience having a light supper here. People, I'm sure, came in from all over the country, and I hope everybody's been jabbed and they are fully vaccinated, and if they're not, that they will get back home so that we can all get back to work, because we really need to get back to work to take care of cancer patients. And so I've had a good time here because both physically interacting with people at the poster session as well as virtually. I gave the William McGuire Award yesterday, and so I ended up spending today while trying to get to posters and sessions, just hearing a lot about, that was a great talk yesterday, that was important yesterday. And so I spent a lot of my time really thinking about what did I say yesterday? And because I was just in a zone, but what's the implication of what I said to what's going on in all the sessions? And I think there's a theme coming on, which is really we're getting a lot smarter about why patients are getting metastatic disease. We're also getting a lot smarter about when you treat a patient and the drug stops working, why does it stop working? And so for a very long time, most of our drug development is when patients have advanced metastatic disease. But I was very interested in going around to look at what else can we do to prevent metastasis. And so a number of really early studies looking at how you can treat patients before you remove the cancer, and then after you remove the cancer, and patients who have complete pathologic response do incredibly well. The cancer doesn't come back, and you've tested that the drugs that you used actually worked in the patient. So that's becoming more and more used now for all the different types of breast cancer, whether it's triple negative, HER2 positive, or even the estrogen receptor positive breast cancer. So a lot of new drug development happening with estrogen receptor positive breast cancer. How about estrogen receptor negative breast cancer? So clearly, we're finding what we call heterogeneity, that I think we've all accepted now that breast cancer is not one disease. You've just heard from Dr. Kaklamani about how different countries are approaching treatment of breast cancer. But all of us are trying to sequence the genes that are involved in driving tumor progression. Either the genes that predispose you to getting the cancer in the first instance, or the genes that then drive the cancer from being totally curable to being advanced and metastatic. And by understanding what all those genes are, and so my work has been really driven by understanding that women get BRCA1 associated breast cancer, and they get what we now call triple negative breast cancer. And then we do lots of genomic assay, and we can talk about basal-like breast cancer, HER2-enriched breast cancer, but our lexicon is getting wider and wider, because it's not just 3 subtypes of breast cancer anymore. It's multiple different types of breast cancer. And so this meeting, there's a lot more emphasis on precision. What does it mean to do precision oncology? What is precision treatment of breast cancer? What is precision prevention of breast cancer? So until you can actually precisely assess the risk of breast cancer, then you may not be able to precisely prevent it. So I was very interested in a lot of the posters and on some of the work looking at the genetic and epigenetic changes that are driving breast cancer progression. And a lot of work now looking at what we call liquid biopsies, right? That was big, and I went to the sessions that are talking about maybe we don't even need to do biopsies anymore. We can just do serial blood draws and then still be able to measure the types of DNA alterations or the type of gene alterations that we can now target. So lots of new compounds that are in development. And then the one really nice work that I listened to this morning was looking at, okay, you started off being HER2 positive, and then all of a sudden you're not responding to all of the HER2 targeted therapy. So do you just march on and go from one drug to the other to the other? Well, maybe not. We're all now taking a pause and saying, let us find out what are the genetic drivers or the metabolic drivers or the things that are really the proteins that are making this breast cancer progress. And by looking at what is changing in the cancer as we're treating it, then we can do better targeted therapy. And of course, targeting the genetic changes in the breast cancer is one thing, but targeting the microenvironment, changing the environment in which the cancer is growing, that's huge. And that's why immunotherapy, all the new studies on immunotherapy, how we can boost the body's immune response to fight breast cancer was all very exciting, and a lot of studies addressing that today.
Okay, we're going to start with questions. So anybody in the audience who has questions, we have microphones you can go to. And we'll also take questions from our virtual audience as well. Stacy.
Hi. Stacy Tignano from Northern California, advocate with a bunch of different people. Thank you guys for being here today. And a question about the CNS involvement that Dr. Kaklamani was talking about. There's a lot of chatter on Twitter about whether or not those patients with brain mets were stable or had active mets. Can you speak to the studies and how it relates to somebody that might have an active brain met as opposed to somebody who has a previous history of brain met?
So, there is a differentiation, so I'm glad you're bringing this up. So the clinical trial allowed stable brain metastases. We still have one clinical trial, and only one that has actively, a large trial, that has recruited patients with active brain metastases, and that's the HER2 CLIMB trial with tucatinib. So it's important for us as clinicians to recognize that, and if we have a patient who has brain metastases that are growing, and we're planning on using a treatment, a systemic treatment instead of a targeted treatment such as radiation or surgery, then the drug that we know has proven to be effective is tucatinib. Now, do I think that TDXD is going to work? I think it probably will, and there are ongoing trials looking at that, but so far the data that we have is with tucatinib.
Hello, Bob Ritter from Ithaca, New York. I have a question about sort of information overload. I'm really struck at this meeting how much information there is now. And as oncologists, how do you keep this all straight? It seems like we're adding more and more and more variables, and I'm afraid your brains about to explode.
We feel the same. To be very honest. No, I think it's very important to be subspecialized, and it's amazing that even if you look here, you have Dr. Olopade. She just primarily prevention, and I primarily do either HER2 or even within breast, we subdivide into our areas of interest, and I think that's how we keep focus. We talk, we understand, but to keep really on top of your field, I think you really need to keep focus, and that's why academic health centers research is important. And to seek help from physicians from those organizations.
Well, I mean, we live in a knowledge age. So I think the important thing is that science is always evolving. And what's really exciting about this moment in oncology is that we're talking about escalation and de-escalation. Right? While we're very excited to have more and more drugs and really have many more options, we also know that there's a concern about financial toxicity, that we can't just wait to add more and more. So even during this meeting, you will hear discussion about de-escalating. How can we be more precise so you get just what you need, right? If you don't need chemotherapy, we don't need to give you chemotherapy. And if you need chemotherapy, we can use lots of real-world data. That's the other thing that, it's sort of becoming really important to the field, that yes, we can't know everything, but we all probably have a cell phone and people can push information to us. And so the question is, how do we get real good data, and what is the level of evidence that we need before something becomes standard of care? So you will hear us being very cautious as scientists that it's not ready for prime time. And then you'll also hear us saying, well, this is really good evidence, it's been replicated, and so it's practice changing. And we're all gonna go home and start making sure that we offer it to our patients. So that's why it's really important to come to these meetings and to engage with scientists and to ask questions and to really make sure that what we know as doctors, that you also know as patients, because you need to be able to have shared decision making. And that's why this session, I think, is really important, that you can ask us questions and you can ask us how we keep up. Most of the time, we don't keep up. But our patients bring information to us and then we go and read up on it. That's the way it works.
Okay, this is from a 2-time triple-negative breast cancer survivor. They would like to know, when will the real deal treatments, which are truly effective, come to all clinics?
Jenny, you want to take that?
I'll take that. I think we have a number of therapies now for triple-negative breast cancer. I think the biggest problem with TNBC is it's a negative, negative, negative. So we're defining a disease based on what it's not, which means no estrogen receptor, no HER2. But we do now, have the approval of immunotherapy. We have approval of ADCs, and antibody drug conjugates. They are effective. I think if you're asking for a magic bullet, I think there is ways to go. But I think by understanding the biology behind TNBC, by understanding the different types of TNBC, and not using one size fits all, but personalizing the therapy according to the subtype of TNBC you have, I think we're making progress. We really, really are. I mean, 3-5 years, we didn't have immunotherapy, we didn't have ADCs, and we had more chemotherapy upon chemotherapy, which may not have been effective. But I think we're making strides. We're definitely making strides in the understanding of TNBC, and also monitoring of disease. As Dr. Olopade was mentioning, we can now do liquid biopsies, we can test in real time, and we can do molecular tumor boards. And with that, I'm going to switch over to Dr. Kaklamani, who will tell you about molecular tumor boards. The second question.
There was a molecular tumor board today. Unfortunately, I didn't have a chance to attend, and I'd love to, but this is why we added it to this meeting, because these molecular profiles that we get are extremely complicated, and unless you have an expert that's all they do, it's very hard to interpret these reports. And each company tries to interpret the reports for us, but we also have to have an understanding. And also, we have to have not just a target, but we also have to have an effective therapy. And that's where Dr. Fabrice Andre's clinical trial that was presented a couple of days ago is so important, that if we don't have the target and if we don't have the drug, any treatment is not going to work. But even if we don't have the target, we still need the effective drug. So these molecular reports, a lot of times, especially in triple-negative breast cancer, are disappointing because we find nothing. But many times they help lead to treatments or clinical trials, and how important clinical trials are, I don't think I need to remind any of you, that help progress the field.
Okay, we'll go to outside. Barbara?
Hi, my name is Barbara Segarra. I'm from Puerto Rico. I was happy to see at least 2 sessions about diversity and inclusion, and it seems like everybody is talking it again, because this started in the 90s and we're still in square one. But happy that it wasn't, we're talking about all these strategies. So my question will be, like, is it realistic? Are we really going to do better including minorities in research? Especially, I talk for the Latino population that is the fastest growing minority. And my second question or comment would be, how do we get all this good information or complicated information to our community oncologists for those patients who cannot get to the academic centers and should benefit from all this knowledge?
I think diversity is one of the most important things that we need to deal with. I think that the days where we, basically divided breast cancer into 3 subtypes, that, as we know, is clearly inadequate. And then I think our knowledge with respect to different ethnic backgrounds and race is also inadequate. So one size really doesn't fit all. And I can almost bet you that as time moves on, when we're older and gray, what will happen is that we will realize that there are different responses, different targets based on race and ethnicity, I'm very sure of it. And that's why it's critical and imperative that we continue to encourage and actually empower women of color, men of color, patients to basically enroll in clinical trials so that we can understand the biology that drives these different diseases, and they're going to be different. And the responses are different, the toxicities are different, and we can see even from the Chinese data, there was a great paper that was presented, it was yesterday, I'm getting lost, by the Fudan University that shows TNBC may be slightly different in the Asians. So having more diversity, we are very Caucasian-driven understanding of breast cancer, which I think has to change. But it also behooves, and I'm very strong about this, that we need to basically, I'm a little bit crude, we need to be able to empower advocates, pay advocates, because it takes time off. Women who come from minority backgrounds have other considerations. They have to put food on the table, they have to take care of children, the logistics of just transportation. I'm very passionate about this, sorry. But you need basically to empower these women to be able to enroll in clinical trials. You can't just give lip service and go, oh, I'm going to have one palmatorad go out and, that's not enough. You really, really need to empower women to basically be able to enroll in clinical trials. And yes, I think we are going to make a difference.
Yeah, so, on Monday, at the beginning of the conference, we had a long discussion about equity and really, trust in science and how do we engage with communities of color and make sure that our treatments work. And just like we've been discussing, we have a human race, and we have, especially as we begin to do genetic studies, right? One of the things that we think about is that we all have our genes, and it's 99.9% identical. But socially, both in this country and every country in the world, we're divided by class, haves and have-nots. It just happens that in the US, color is very, very important. But after my talk yesterday, I had a contingent from Peru that came and said, if you go to South America, there are many different countries, and they all have different populations. But when they come to America, they all become Hispanic, right? It couldn't be farther from the truth, right? So how do we get to the granularity that we're talking about, where it's not black or white or Hispanic as just one fell swoop? Even when you talk about Africa has so many countries. So the diversity of the human race is what actually gives what we call survival of the fittest, right? As we begin to have evolutionary biology and so many things that we still need to understand. So when we begin to use genetics to understand breast cancer, which was the point of my talk yesterday, is that we're going to find so many things that are different, but then there would also be many, many things that are the same, right? And because they're the same, they should work well for everybody, right? But if we have divided the world between haves and have-nots, even the things that are supposed to work will not work, right? So that's the job of all of us as advocates, is not to divide ourselves up, but to now say, you know what, we're in this together. And so I always like the example of the AIDS epidemic. It was an infectious disease. Everybody came together and they acted up and they got clinical trials. They got lots of money to try and eradicate HIV/AIDS. And that battle and that continues to happen. And now the disease that was definitely a death sentence, now people are living longer and longer with HIV/AIDS, and now they're getting cancer and we're having to treat them. So the same thing, if we could really come together, and that's why I like the cancer moonshot, and there are a lot of things that are happening now as a result of all of us saying that we can end cancer, but we need solidarity, and we need to begin to use all of the tools that we have available. And it so happens that we know a lot about genetics. We know now about RNA because we now have RNA vaccine. And now with RNA vaccine being, taken from cancer research to go and fight this pandemic, we had sessions on COVID and what this means for the cancer patient. So what's going to happen after the pandemic? All of these scientific discoveries, how are we going to bring it to actually helping every breast cancer patient? So I'm very excited that at the end of this, we will all come together because now we recognize the inequities and the really poor outcomes that are due to the fact that we have not paid attention to those who do not have. We have not broadened access to clinical trials, and then we blame patients for not participating in clinical trials. So the onus is on us as researchers, as cancer centers, as pharma, to take clinical trials to where people live. And so in the way we design clinical trials and in the way we engage every patient with access to clinical trials, I think we would make much, much faster progress than we have done up to this point, where we have you drive to wherever you can to go and get access to a clinical trial. It shouldn't be that hard to get access to a clinical trial.
Mm-hmm. Next question is, are there any updates on treatment of leptomeningeal metastases or any trials that accept these patients?
So leptomeningeal metastases is where the cancer has seeded on the leptomeninges, which is one of the membranes around the brain. It's probably one of the most aggressive presentations of breast cancer. And it's one of the most difficult kinds of presentation to treat. There are, most of the trials, as the advocate is suggesting, do not include patients with leptomeningeal disease because, unfortunately, the survival is very poor. But there are trials now, and they're looking at tucatinib, and there was one presentation yesterday at one of our sessions about that. And I know they're also doing trials with T-DXd on leptomeningeal disease, so we're becoming a little more brave. Probably our drugs are getting better, and that's why we're more brave in including patients, because we do need to. We lack very good information on what to do with these patients. We've been treating them with very high doses of chemotherapy and trying to really get to the leptomeningeal compartment, but there's really not very good efficacy so far. I think tucatinib and T-DXd for HER2-positive leptomeningeal disease are going to work. For the non-HER2-positive breast cancer, that's an issue. We have some trials with another antibody-drug conjugate called sacituzumab that might show some improvement, but it's very early.
Okay, we'll go back to the audience again.
Hi, I'm Sandy Finestone from Southern California. I just want to build on something that Barbara said. Patients who come to you are extremely fortunate because you are the clinicians that come to these meetings and you work on these cutting-edge treatments and you're so knowledgeable and not just that, but you're so caring. And we as advocates are also privileged to come to these meetings, and we take what we learn back to our communities. But our reach is quite limited to other patients, to women and men in our communities. But we have, at least I'll speak for myself, I have great difficulty reaching the community clinicians about patient needs, about patient concerns. And I don't think it's my job to educate them. I couldn't anyways. So my question is, within your professional organizations, is there a program, is there a discussion about reaching these community clinicians to ensure that maybe they don't have access to some of these cutting treatments, but are they aware of them? Are they aware of the trends? And I'll give you an example. I sit at a university in my area. I sit on their breast committee. And one of the clinicians brought a trial with adriamycin and another treatment modality. And everyone said, oh, this is great, let's do it. And I raised my little hand in soft voice and said, why are you treating with adriamycin? And the clinician said, what's standard of care? And I questioned it. But if I hadn't been in that meeting, that trial would have gone through. And I don't know what else to do, but I guess I'm asking you, is there a congenial, is there a professional platform that looks at educating community docs about these kinds of things?
I think your concern is extremely valid, and one of our roles as academic physicians is to educate. And we don't just educate medical students and residents and fellows, but we educate our community. And I think the pandemic has actually helped, because now you have all these virtual programs. So you don't have to travel to go and listen to somebody, or you don't have to travel to give a talk to someone. You just sit in your office or your home or your couch or your bed, and you either talk or you put one of those virtual backgrounds. Nobody knows that you're laying in bed, and you can talk, or you can listen, and so I think, and this is one of the strengths of having a pharmaceutical company do these talks, because obviously they have the resources, but we have a lot of CME companies. They're doing advisory boards, and they're doing advisory boards for community physicians. It takes time, and some of these physicians may not be interested, and at some point you're not going to have a 100% success rate. And I think that's why I always tell patients they have to be their own best advocate. And I understand that socioeconomic issues might not allow them to be, but they cannot just depend on the physician. Like Dr. Olopade said, I've had many patients that come to me and say, what about that treatment? And I think about it and I go, you know what, you're right. And I change my mind because they were right. And I love that. I love that dialogue.
I think I have two comments. I think it behooves San Antonio Breast Conference to do CME in a new way, to be able to get to community physicians. It can be podcasts, it can be short things, but updates, highlights. I think it's a new world. And in the same vein, I think the pandemic has actually shown the way that we can actually practice medicine differently. Why can't we do video visits as a one-off?
Hi.
Center. I mean, all of us would love to give input and help more women. We don't have to be physically infusing the chemotherapy. That's not, frankly, I don't care. I would like to, make sure that the treatment is in the right track and stuff like that. I'm sure you're the same. So now with the video visits, why can't we do that? So I think the pandemic, we're changing. I hold your breath. I think this it's been a terrible time for everybody, but there are some glimmers of hope, and I think the way we educate and the way we treat patients may be something different in the future.
And the video visits, Medicare is allowing them. They just gave us an extension. We're hoping that they will make it permanent, but this is also an area where advocates' input is extremely important. I have several patients that now they have a treating physician in Austin, and they have me in San Antonio, and I talk to them every once in a while, and we have that connection, and I'd hate for them to drive 2.5 hours to come and see me.
One from our virtual audience. To all the panelists, since neoadjuvant chemo gives you the answer to drug efficacies, even without PCR, subsequent surgery and radiation, can you give a good result? Discuss.
Yeah, I think I like that one. It's not one, again, it's not the same for all. So the most important thing about neoadjuvant treatment as a whole, it tells you whether or not that treatment that's been chosen for you is effective. And if you don't get a PCR and you have HER2 positive disease or you have triple negative breast cancer, there are now new treatments, now actually ER as well. So for all different 3 subtypes, whether you're ER positive, whether you're triple negative, whether you're HER2, if you do not achieve a PCR, we know that you have a slightly higher chance of relapse, at least for the TNBC and the HER2 subgroups. And we have new treatments that can improve that outcome. So that's the reason why I give neoadjuvant chemotherapy or neoadjuvant therapies. And trials.
Yeah. So I talked a little bit about the iSpy network, which really at the time that Laura Esserman and the group really started talking about it, it was designed to be able to bring investigational new drug treatment early on to patients who have what we call high-risk breast cancer. So locally advanced breast cancer that's high risk and has a high risk of recurrence. This is really a very nice spot for all of us to think about new drug development now. So the exciting thing is if you get to a path CR, and now we have Chuck Peru also presented data from Alliance where they use different combinations of drugs, adding immunotherapy. It doesn't matter. If you give treatment upfront and you get to a complete pathologic response, you're good. You feel that you have responded to the treatment and there's a chance that the cancer would not come back. But then how about those who do not have a complete pathologic response? What can we learn about mechanism of resistance for those patients? And I think if there's any one area where I am passionate about, it is that we then pause and we use that opportunity to ask, what has changed? Why didn't all the cancer cells melt away? Because even if it's just one cancer cell that remains and it's now become resistant, that might be the one that then grows back and then becomes metastatic. So there's a lot of interest now to try and figure out what is remaining, and if we can find whatever is remaining and why it became resistant to the treatment that we use, then maybe we'll be able to develop a better way to avoid that, overcome that resistance. So at this meeting, lots of posters, lots of presentations about the mechanisms of resistance and then developing a different approach to overcome that resistance. And so some of the pathways that we hadn't thought about before, and I think one of the questions that somebody asked was, there's so many things that can be mutated or that can be abnormal in a metastatic breast cancer. Were they always there? Or, for example, the estrogen receptor can mutate. And if the estrogen receptor mutates, then you're not going to be responding to your endocrine therapy. Well, we learned today that if you find that and you change treatment, then you can prevent that cancer from the mutated cells from becoming the dominant clone. So really lots of reasons to be optimistic that we now have the tools to monitor in real time when patients are becoming resistant to therapy, and that after neoadjuvant therapy, if you do not have a complete response, then we have an opportunity to, for example, if you have a BRCA1 mutation, well, maybe that's the time to switch you to a PARP inhibitor, and now we have oral PARP inhibitor that work in the adjuvant setting. Well, maybe we should be doing all sorts of different clinical trials to bring these oral drugs, Tamoxifen is an oral drug. You can take it. So we've used it for adjuvant therapy for 5 years. Well, now we have a PARP inhibitor. We have capecitabine. These are all oral drugs that you can use for triple-negative breast cancer. And they are making a difference. There was a big announcement about the selective estrogen receptor modulators, SERD and SERMs. They're all oral. So all different types of oral medications now that we can develop in the pipeline to convert breast cancer into a chronic disease that you just take a pill for. So I think these are all lessons that we're learning from neoadjuvant therapy, neoadjuvant approach to treatment, and then extending it to find early examples of resistant clones that we should be targeting.
Hi, I'm Sharon Anderson. I have inflammatory breast cancer, triple positive, de novo metastatic. My question is, when you have IBC, you get disqualified from a lot of clinical trials. And I feel like we should be included in clinical trials and be able to offer what we have. There's a lot of us that are exception. We're long-time thrivers. That's something to look at as well, in some type of trial to see what is the difference and what can we offer for the clinical trials. But getting disqualified because I have inflammatory breast cancer, I just don't get it. I don't understand it.
You're absolutely right. Now, there are physicians that believe that IBC is a different subset, and that's one of the reasons why we disqualify patients from trials. The second reason is because IBC is aggressive, and the same thing as with leptomeningeal disease, we do not want to take the risk of accruing patients that have very aggressive cancers because we're not brave enough thinking that our treatments are that good. So that's a problem. But there is a thought that IBC is a different kind of breast cancer, and I don't think that we truly have the data to conclude that, but people are working on that, and it might end up being in its own entity, and that's why there's clinical trials just for IBC. And those are going to be in the large centers because obviously IBC is relatively rare. But I think you are right, and I think that especially in the metastatic setting, there's no reason why we should be excluding any IBC patient, because once the cancer metastasizes, it's aggressive, it's metastasized. And so all of these cancers that are metastatic are aggressive, and we have to find the best treatments for them.
I just have one more thing to add. I feel that, if I run out of options, there's nothing. There's no data for me.
Yeah. So actually, the question about inflammatory breast cancer gets us to how many different types of breast cancers are there, right? We were just talking about heterogeneity and the fact that you may think that the inflammatory breast cancer that you have is rare, but we have data that it's actually not rare in Egypt, for example, and there have been international consortium actually studying inflammatory breast cancer and finding out whether there are other ways that we can treat patients like that. The problem is we don't all share the same information as a global breast cancer community, so everything becomes rare, right? Yeah. But it's also the case that because something is common, and we're also all beginning to ask ourselves, when you say, I have estrogen receptor positive breast cancer, it can be 100% positive, it can be 20% positive. So not all estrogen receptor positive breast cancer is even the same, right? So we're now beginning to get to this age where we're even looking at the different single cells that make up a breast cancer. So we're sequencing what we call the gene expression of every cell in a breast tumor. How are the T cells, the B cells, the macrophages, the fibroblasts, then the epithelium, what is breast cancer? When what you actually see are all sorts of things that form that lump that is called breast cancer. And how are they all connected to one another? So there's a whole new learning, an whole new way of looking at breast cancer that we're saying it's becoming even more complex and more challenging to put it all together. The good news is we're beginning to use artificial intelligence and deep learning and technologies that are actually dependent on big data. So you will hear people now talking about big data, that we need to pool real world data. We need to find that one person in this country, in that country, and then bring all the data together so we can now start forming real numbers and critical mass to target what we call the rare breast cancers. So I feel we hear you and we know exactly how lonely it feels when you feel that you don't have the type of breast cancer that everybody has. So who is studying inflammatory breast cancer and what might be some of the options? So a lot of the things that we're hearing now is that it may not even be necessary to talk about breast cancer, lung cancer, prostate cancer, that it may be that we just need to know what drugs work for what pathway. So if it turns out that the cancer has DNA repair that is damaged and we have a drug that targets DNA repair or that targets the RNA, then let's just use the drug for the cancer and not worry whether we call it inflammatory or lobular or ductal. So keep coming to San Antonio and keep your eyes and ears open, but there's really a lot of new ways of looking at old diseases that we didn't know how to study.
And there are new drugs coming out, so don't lose hope.
Ranch, and I am with the UNC Breast Spore at Lineberger Cancer Center in North Carolina. When you were talking about the new drugs, the SERDs and everything, I am concerned about the cost of these drugs and access for patients. And have you had any resistance with patients not being able to get the payers to pay for the drugs for the patients? Because this is a real concern. These drugs are costing thousands and thousands of dollars. And so are you getting questions like that from your patients? And, are they able to get it paid for?
It depends. Most of the companies, or actually all of the companies, have programs to try to help patients with copay assistance, so they are doing the best they can. You're correct, the drugs are expensive. A month of one of these drugs is over $10,000. But obviously the patient doesn't pay $10,000, the insurance doesn't pay $10,000. And somehow it gets covered. The copays can be high. And it also depends on what insurance a patient has. I think what I've learned is that it's better to have no insurance than have crappy insurance. Because if you have crappy insurance, then the drug companies' hands are tied. All of these programs out there that have been put in place for the uninsured, you're not eligible for. If you're uninsured, then we're usually able to get you on one of the programs and kind of help out in many ways. And if you have good insurance, obviously, many times. But the financial toxicity, and Dr. Olopade mentioned that before, is a major issue. And the patient's financial toxicity is something that we all need to care about. Medicare, one of the issues with Medicare is the way things are is it makes patients ineligible to receive a lot of support from the drug companies. So that's why the companies are going to talk about copay support for the non-Medicare patients. And obviously we know that the median age of breast cancer is 62. So many of our patients are Medicare patients. So there's an issue, and if they don't have supplemental insurance, then some of the copays might be might be high, and that's a major problem. And that's yet another opportunity. I think what we're all learning is that every problem also equals an opportunity. So an opportunity for our advocates to go out and advocate for these things, and they're the ones that are dealing with this more than we are, and they need to let the government know.
Can you comment on the Emerald study, and what about patients who've already been on fulvestrant and CDK4/6 inhibitors, and also what if you've had prior oral chemo?
So I guess since I was one of the co-authors, I'll answer that, but I'll be happy to hear my colleagues' take on the results, because I am definitely biased when it comes to that. So elacestrant we studied it in a phase 1 trial, so that's one of the very early trials where we're trying to figure out what the right dose is and make sure that this is not too toxic for our patients. And then obviously the Emerald trial, which is a phase 3 trial. And in both of the trials, actually, especially the phase 1 trial, we had many patients that had been on prior fulvestrant, prior CDK4/6 inhibitor, prior chemotherapy. On the Emerald trial, that was allowed as well. And we know that elacestrant is active in that patient population, but you saw the activity. It's not huge. So we have a lot of work to do. All we proved with the Emerald trial is that it's probably if not the best, a very, very good endocrine therapy option. But we also learned that we have to do better. That when you look at the curve dropping within a month and a half on 50% or so of the patients, we have to find who that 50% is, because it's unacceptable from now on to offer those patients endocrine therapy by itself.
I was very excited about the study.
Well, thank you.
We waited 20 years for a new
For something new, right?
Something effective, new oral. And, you know, I know the comparator arm could have been better, but it's a new SERD. You don't have these big, fat, painful IM injections of Fascidex. I was excited. Sorry, I'm online now. I can say that. Well, exciting new therapies in the pipeline to treat HER2, HR, triple positive, triple negative BC. I'd like to take that. I think this is very interesting. There are certain areas we need to escalate therapies, and there are certain areas we need to leave it. Don't take it away. I can't remember. Oh, de-escalate therapies. And so, for example, HER2, we have heard from Dr. Kaklamani. We have exciting new drugs, especially the ADCs, and there are more ADCs coming down the pike. It's just going to be stunning. And ADCs flummox biologists because they actually work in ways which is not just targeting the HER2. All you need the target for is to inject this toxin and kills the cancer cell. So it flummoxes all the years of cancer biology that we have done, which is a good thing because, you know, we have new tools, and I think HER2 is a very exciting new area that especially HER2 positive, HR negative cancer. And I think that for triple positive breast cancer, where the discussion was, is that we are now moving to the idea maybe even in these patients do really well, especially if they get a PCR. There may be actually ways of de-escalating. Are there patients that we do not need to give chemotherapy to by looking at what the targets are and doing neoadjuvant studies to see whether or not we can achieve a PCR. So I think that looking at these new treatments, there are ways in which we can escalate with ADCs, we can de-escalate treatments. And in the patients who don't get a PCR, I think these are opportunities to figure out new treatments, and there are new treatments. For ER positive, for HER2, for HR positive breast cancers, I think the SERDs and the POTACs and all these guys, they are coming out that degrade the estrogen receptor. These are very exciting new drugs, and I'm really, it's been a long time since we've had new drugs in the hormone receptor positive platform and arena. So both ways of upping treatments for those that need it and de-escalating treatments for patients who do not need it based on your biology. And there are a lot of new treatments.
HER2 low?
HER2 low is exciting. So basically, it's all about these so we have. Why did I get my PhD? We got a PhD to understand cancer biology, and we found all these targets. We found out how it works and all the mechanism, and we learned in all these cell systems how it works, and then it doesn't matter because all you need those targets for is a way to, it's true, it's really sad, but it's good. It's a way of targeting and engaging the target with a toxin. That's how ADCs are working. And Dr. Kaklamani was being evil there, because in the HER2 low, HER2 is not the driver of the cancer, but can form a way in which the toxin can get into the cancer cell. And watch the space, it may be effective. In early studies, it looks as if it may be, but we're still waiting for the definitive studies.
Yeah, and I think, one of the things that you should be on the lookout for is all the things that we thought we knew that we actually don't know. So this whole idea about HER2 low and what is it that we're targeting? Is it the tumor? Is it the microenvironment? Are we causing inflammation, and then inflammation is recruiting immune cells into the tumor and killing the cancer cells? All of these things are now they're complex, and the more we learn about them, the more we even get more confused about what we thought we knew before. So it's not that patients don't trust us or that we're trying to deceive patients. It's that knowledge and science is really exciting, because who would have thought that every year Apple will get me to buy a new phone because the camera keeps getting better and better. And as soon as they come in and they say, oh, we have a new thing, I want it, right? So that's what happens to scientists. We get new toys and we start playing with it, and then suddenly we find out, wow, this is so beautiful. We need to translate it in the clinic, and it takes a while. But the rate at which all of this is happening now is just phenomenal, just because we have the tools to do it. So, when we first started sequencing the genes, I can tell you it would take you 6 years, and I know this because I was trying to sequence just 2 megabases of the chromosome when I was pregnant. And then we found the gene when my son was in 1st grade. So that's how long it took us, right? These days, you don't even need to go out of everything is on your computer, and you just need to align the human genome that you've just sequenced on a reference genome on your browser, right? And so a lot of the discoveries have now been made in somebody's basement, just aligning genomes and trying to make meaning of the sequence. So, and then bringing in new powerful tools to even understand what all of these things mean. So I think it's exciting that, cancer biology, cancer genomics, we have new fields, coming into medicine every day and bringing in fresh ideas about how to discover new drugs, how to make old drugs work better, and then how to reduce toxicity. And I think that's why all the different drugs that are now in development, 5, 10 years from now, the kinds of drugs we're going to be using to treat breast cancer or prevent breast cancer will be very different. And we're going to be looking back and say, oh, that was so barbaric, what we were doing.
I totally agree, and I think one of the things which I wanted to highlight a little bit about is, you're talking about an immune landscape. We now have immune checkpoints, but that's not enough. We have new ways in which we can alter the tumor microenvironment. That's one of the questions. And, change the inflammatory background, the backdrop to the cancer, so it allows these cancers to grow. We have a paper coming out in Science Translational next week, actually. By using a repurposed drug, we were able to change the tumor microenvironment and improve response rates, actually double the response rates in metastatic triple-negative breast cancer. And the tools that we have to look at the immune landscape, it's phenomenal. So it's a drug that targets inducible nitric oxide. But we're going to have, as you rightly say, Dr. Olopade, we're going to have all these new ways in which it's going to be different. It's not just going to be chemo. It's going to be microenvironment, your microbiome, your immune system. It's just going to be a whole new way of looking at cancers. I'm really excited. And because we understand the vulnerabilities of cancer through all the work that we have done, I think we will have new treatments and less toxic treatments.
Hi. I actually just have a request. And I've heard the word de-escalation a bunch this year. I heard it a bunch in 2019. I've had conversations with individuals, I don't think specifically with any one of you. That word is loaded. It's loaded in the same way that the word prevention is loaded. Oftentimes when we say prevention, what we mean is risk reduction. I know in public health we have primary, secondary, and tertiary prevention. That is not what the general public understands. When we talk about de-escalation, what patients hear is, you're not going to give me everything you can. And that invokes fear. And so I've humbly suggested to some people, Dr. Miriam Lesberg has picked up the gauntlet with me, to talk about right sizing. You're personalizing the treatment, you're right sizing the treatment. It's a comfort. It makes people feel seen, heard, and held. And I just want to offer that as an option.
Thank you.
Thank you.
Very
much.
Yeah,
very
actually.
I like that.
Well
yeah,
said.
Well, you know, these are nuanced discussions that we're talking about. This is where I wear my global cancer expert hat. You know, we're blessed in America because everything is big in America and we want everything. And we want to personalize it. And if we have it, we never want to share it with anybody because it's my personal property. That's a mindset that is only in America, okay? Because San Antonio is an international breast cancer meeting, I like to think about the perspective of women who don't have anything. We're not even talking about de-escalation. We're talking about increasing access. So somebody just has just a little bit of what we have.
I am
Okay?
I love that term. It's just the term de-escalation.
Right. So when people are talking about escalation, right, it's that we waste so much of our GDP. We spend 17.4% of our GDP. The number one reason why cancer patients go bankrupt is because of the treatments that we have. So when I started my training as an oncologist, we were doing bone marrow transplant for breast cancer. And breast cancer advocates were pushing that we should transplant everybody. And we have to be humble enough to know that we did that. And maybe by transplanting some women, we actually accelerated their death, right? So I always like scientists to be humble about what we know and what we don't know. And so there's a lot that we don't know. Absolutely. I hear you about escalating and de-escalating. And I think it's really because of what you just said, fear, right? Our job really is to provide more supportive care, more psycho-oncology. You can see we're all medical oncologists. We're excited about more and more drugs, right? But maybe if we paid attention to how can we communicate with you so that you're not afraid, right? So words do matter. And I think that for us, we have to learn how to communicate better. So I do risk assessment. And I said in my talk, in 1996, when we had BRCA1, and I would say there's a high chance, probability, that you would get breast cancer because you have BRCA1 mutation. And if we can model it and we can say, here's the chance that you get breast cancer. And at that time, we will give you an estimate of, it's 87% chance that you're gonna get breast cancer. But we were also humble enough to say, we don't know nearly enough, right? And by now, we're saying, maybe it's not 87%. Maybe it's only 40%, it's 50%, you know? Virginia and I do this, and we've seen so many families. My oldest BRCA2 mutation carrier is 100 years old, right? 100 years, celebrated 100 years. If we had known that she was a mutation carrier, I don't know what we would have done to her, right? So I'm always very humble to know that there's something we know today. And so what you said about prevention, there are a lot that we know about prevention, but because of the way the language is received by people, there are so many advocates who don't believe we can ever prevent breast cancer. Because the lived experience of the advocate is that I have breast cancer and don't tell me about prevention. So we're all now coming back to the continuum of care. What do we know about the normal breast and how that breast becomes transformed to become cancer? And then what do we know after we have finished treating that cancer about supportive care so that you recover? And I heard somebody say, I'm a thriver. You know, we didn't used to use the word thrive when I started. We used the word victim to talk about being a breast cancer survivor. But now, young people and more people are saying, I want to be a thriver. It doesn't matter. So I think words matter. So I hear you about de-escalation and escalation, but I think that, you know, there's so many things that we need to be able to discuss openly and honestly among ourselves as advocates as well as scientists and doctors. And we don't all have the knowledge that we should have. And hopefully over time, as we do hypothesis-driven research, as the other times we were talking about community doctors. How long does it take from when we discover a new gene or a new way of doing things to when that discovery actually makes a difference in somebody else's life? Billions of dollars to do clinical trials to actually make a difference. How do we accelerate the billions of dollars that we use to get one drug approved for one indication? So I think the community, as we're thinking about it, we also need to start thinking about financial toxicity. So that's why oncologists are talking about de-escalation, that we don't want to cure you and then have the legacy of having debt for your family and people going bankrupt. So it's a real problem. It's a real challenge for us, and we're all challenging ourselves to think a little bit more broadly. We were talking about, as part of this, depending on where you are, you either like Build Back Better or you don't like Build Back Better. But Build Back Better was actually started when, after the war, the Japanese coined it, that we learned from the mistake of having been in a war. Let's rebuild our country. And when we rebuild, let's build it in a way that brings us all back together. And so there's a lot of things that happen, and everything we think about is within the political context, within the country context, within, the community context. But, we're one breast cancer community that is broader than each one of us. And that's why I like to think about the global community that's also looking at us and thinking that we waste a lot of money. In the NHS, you practice in the NHS. In Europe, you practice. There's so many drugs that are not even approved that we use here. We take it for granted.
They haven't even been submitted because they know ahead of time that it's not going to be worth it. They're going to be disapproved.
Hi, I'm Denise Honte from Dallas, Texas, and a proud metastatic triple-negative breast cancer thriver. First-time attendee, excited to be here. The most exciting thing that I'm seeing, yes, is a lot of research, but I also see that there is emerging technologies and the liquid biopsies and the biomolecular imaging. How do we get the word out to the doctors to sign up for these things that redefining NBC, the company defined NBC and Natera, those types of companies, the circulating tumor cells, and taking a look at ctDNA, how do you get more doctors to actually sign up for it? Now, I will say I am biased. I have a phenomenal doctor that will sign up, but I'm worried about the other people that wouldn't. And I'll mention her name, Dr. Joyce O'Shaughnessy. So.
Yeah. Sign up. Okay.
So there's a lot of things we know, but there's even more things we don't know. So it's very important that what we do in clinical practice is something that's been proven. Now, clinical research, and you're right, Dr. O'Shaughnessy is a leader of many clinical trials, and I encourage all of you to participate if the trial is right for you, because this is how we learn. So as part of clinical trials, we do ctDNA, we do CTCs, but what we learned was from our failures. For example, CTCs, we found that you identify them and it's great, and we all feel like we're doing something, but ultimately, the overall survival of our metastatic breast cancer patients didn't get any better by following what the CTCs were telling us. Now, part of it has to do with the limitations of our treatment and the fact that the CTCs were not measuring a specific target. So things are changing, but until we find that what we're doing is improving the lives of our patients, either improving quality of life or survival, then it's still research. So be very careful.
Yeah. So one of the things that I don't know if everybody still swears to the Hippocratic Oath. And now, when I'm teaching in my clinic, I always tell everyone around me, first do no harm. Right?
Non notary, say it in Latin. Prima non notary.
Yeah.
It was Greek.
It was
Latin.
Well, we don't even know whether it was
Hippocratic
Greek or
Oath
Latin.
was Greek.
Sorry,
But it's the language that we all should understand. First, do no harm, right? And so by nature, doctors should be conservative because we should be humble enough to know, and this is what my patients tell me on the south side of Chicago, you're not God, right? So I have to know that I can only promise you what I know. Let's try this, right? And so if your doctor is not comfortable with a particular tool, they have a right to be able to think about it before they offer it to you. So I would rather err on the side of caution, but then because we're researchers, we always say, sign everybody up for research. The first thing I want to do after I leave this place is to figure out how I'm going to get a huge study going to do all these tests so that 10 years from now, when somebody takes over my practice, they will have a longitudinal data set to say, this is what Dr. Olopade did 10 years ago, and look at what we learned now, right? Because what we did in 1996 with BRCA1, we had no idea whether we were going to help anybody. And Werner is here when we had, we called lawyers, we called doctors, there was not even HIPAA, there was no privacy law, there was fear of genetic discrimination. And we all said, this is too important not to study BRCA1. Now, how many PARP inhibitors are in clinical trials? If those brave women had not said, sign me up, get my family tested, let's study this, we would still be fighting whether BRCA1 causes breast cancer or not, right? But we did it. There were brave women and families, and we saved a lot of women from dying from ovarian cancer. And that's how patient-oriented research moves us closer to helping everybody. Are there some mistakes that we will make along the way? Certainly, as long as we learn from our mistakes. And so that's why I think first do no harm is what I write on my forehead when I go to clinic every day.
I'm going to tell you a joke. A patient of mine said MD doesn't stand for minor deity.
Minor deity.
And it's true, it does not stand for minor deity. We're very human.
So with that, I mean, how do you push the doctors to sign up? And I can appreciate that there's some caution in signing up for everything, but I absolutely believe and agree that this needs to be a patient-centric, patient-driven environment in healthcare and the way that we need to go. And I'm a patient advocate, and how do I get the push out to doctors to try new things? And especially if you have, since we're concerned about the financial access to it, like you have the Define MBC, or you've got the new PET scan imaging, the Serana that you can do, but how do we get the doctors, more doctors to buy in?
Well, I think the companies would also need to go figure out how to get this test done faster, cheaper, right?
Yeah.
The innovation is what we all want. It should be faster, it should be cheaper, and they should be out there signing up doctors in everywhere. They shouldn't just go to the fancy doctors' offices. This is what I'm saying. They should come to the south side of Chicago, put it in the toughest neighborhoods, and get me data from the toughest neighborhoods. Then I will know that you're really serious about making it accessible to everybody.
I think we should push the companies to have corporate shared responsibility. They should be do one, give one. I mean, you want to get it out, you make the companies bring the prices down.
Do you find NBC, I think it has an early access program that's offering it for free.
Yeah,
So
that's
why not
okay.
do it?
Sign up the doctors.
Get up all the doctors.
Yeah,
Sign
because
up.
one of the things I find in big cities is that the doctor said, we don't even have resources to participate in clinical trials. We want to do it, we'll sign up our patients, but nobody asks us.
I may not have been at any of the sessions. I wanted to see your thoughts, or if you could talk about proton therapy, any kind of targeted therapy, that if you believe in that, if you have that person that had that one little rogue cell that got out before being metastatic, or even after being metastatic, if it's that area. What are your thoughts on proton therapy?
I'm not touching.
I'm going to be honest. I think for breast cancer, it's not. I'm not putting my head out there. It's okay. I'm not a believer in proton therapy for breast cancer.
No.
It hasn't shown any efficacy so far. Doesn't mean we shouldn't be studying
Doing
it
it,
more.
right.
It doesn't mean that we haven't found the right patient population, but right now it's
And if you're talking about financial toxicity, that's
Yeah.
going to be
Welcome
one
to the financial
very, very
toxicity.
toxic treatment in terms of financial toxicity, not in terms of.
Oh, thank you.
Could you comment on the use of lower dose chemo and the opportunity to find doses that may be effective while having significantly lower profiles?
So I think this is one of the limitations of how we now do phase 1 clinical trials, which is finding the highest possible dose before the patient dies. And that is not the way we should be doing phase 1 clinical trials. The way we should be doing phase 1 clinical trials is finding the lowest dose that is effective. And that takes more patience, that takes more time, and that probably means a little less money for the pharmaceutical company because the drug is going to be used at a lower dose. But I think that's the right way to do these trials. And we've discussed this before, where instead of just looking at how toxic your drug is and let's just push it to where it's really toxic, but it's just acceptable, let's find the target, which we know because all these drugs have targets, and let's see how the target gets turned off or on, depending on what you're doing, at the lowest possible dose of the drug, and then give the drug at that dose. And all of these companies are afraid because they're thinking, well, what if it's not the most effective dose and maybe we should go up? But in most cases, that's not what happens. And I think the person that wrote that question is absolutely right. We should be doing this.
Yeah, so the one comment here, about the how we set doses, and even this question about the PCR, why are doctors in communities not using neoadjuvant therapy? Do you know?
I don't. I think they should be for triple negative and HER2
positive, we should.
I mean, the right sides, obviously, but you definitely should be, because by not achieving a PCR, it makes a difference, and we have new therapy. So it's out there, and patients you're your best advocate. And if if you have triple negative breast cancer and HER2 positive breast cancer, you should be receiving, by and large, the majority should be receiving neoadjuvant chemotherapy. Agree?
Agree.
I think even for now with the SERDs and the new way to treat estrogen receptor positive breast cancer, I would say for every type of breast cancer, you should be using neoadjuvant therapy now. We have lots of data about that, and you can learn about the patient.
Absolutely.
But that sometimes old habits are hard to break.
Yeah.
I think it's because patients get referred to surgeons first in the community. rather than medical oncology. So, and then the surgeon goes, old-fashioned, we'll just take it out. So that's the mistake.
Yeah.
One more question from the
Thank you so much.
audience.
This is my first time here. I'm Daniel Cook from Symbiosis. One of the themes of this conference to me has seemed like stratifying patients into more and more small subgroups so that you can find the right therapy. Can you comment maybe a little bit about how clinical trial designs are changing to be able to handle smaller subsets of patients?
I can take one. One of the ways, you're absolutely right. You have to subset because that's the right thing to do because there are different biologies. And I think ways in which we've been innovative is to use the neoadjuvant platform. So PCR is a very validated now surrogate endpoint for HER2-positive breast cancer and triple-negative breast cancer. So we have very strong surrogates. And so we can use, instead of thousands and thousands of patients, we can use a few hundred patients, and we will know whether or not that regimen is effective. So ways around it is to use neoadjuvant platforms. And in the future, in the metastatic setting, we might be using liquid biopsies and different other ways of monitoring response so that we can cut down the size.
And the more targeted the patient and the target, the better the efficacy. And so you need fewer patients because you're going to find a bigger difference.
And I think in this age where we are really thinking more about pathways and a DNA repair pathway and a drug that blocks that works for breast cancer, pancreatic cancer, prostate cancer. So, but they may work in a tissue-specific manner. So I think the onus is on pharma and anyone who wants to bring a drug to market to do a lot more work to know what the target does and then who to target the drug for. And I think the more preclinical modeling can be done, the faster we can do drug development. And then it's biomarker that is linked to whatever drug you want to bring to market.
I want to thank our phenomenal panelists, Dr. Chang, Dr. Kaklamani, and Dr. Olopade. They did a wonderful job.
Okay.
I also want to thank our sponsors because we could not have done this without them. Again, Exact Sciences, Genentech, Sanofi, Genzyme, GE, Pfizer, Viatris, Seagen, Daiichi Sankyo, Eisai, Merck, Grail, Synyo, Agendia, and Cybermedia Productions. And again, thanks to all of you for being here and also to our virtual audience as well. I hope you enjoyed it, and I'm sure I learned a lot. Hopefully you did as well. Thank you.